So yeah, the basic idea of photosynthesis, you're taking sunlight and you're using sunlight to make sugar. And when I say you, not you, but plants, right? Plants use the energy of sunlight to make their food, which is sugar. Now, in order to make this sugar, we have to take light, and you're taking light plus water, H2O, plus carbon dioxide, and you're going to make that sugar. In order to combine those three things to make that sugar, you have to have energy to do that, right? If you're taking more disorder, these individual molecules, you're going to build them up into a larger molecule, you have to have energy to do that. The energy to build that sugar comes from ATP and NADPH. Now, to make ATP and NADPH, we need to use the energy of sunlight in what are called the light reactions. And that's what we talked about right before the break, where these are going to happen in the thylakoid. And basically, we use the energy of sunlight to then add hydrogens to NADP plus and add phosphates to ADP. That's really the basic thing that you're doing in the light reactions. Okay? And in the process, as a technical waste product, you're going to get rid of oxygen, which is beneficial for us for cellular respiration and for plants. Plants will also do cellular respiration. And then what we're going to focus on today is the Calvin cycle. How do we use the energy stored in ATP and NADPH to then take a bunch of CO2 molecules and through a process called carbon fixation, basically link up all of those carbon atoms together to make your sugar. And whenever you see like the formula of photosynthesis, you usually see glucose as your final sugar. Technically, we're not making glucose directly from the Calvin cycle. The sugar that comes out from the Calvin cycle is G3P. And then G3P through some other reactions can go to glucose. And the Calvin cycle is going to happen in the stroma of the chloroplast. All right, so that's kind of a little overview. I wanted to go through the light reactions as quickly as I reasonably can. A lot going on here, but let's just kind of walk through this story. So here's our sunlight. Our sunlight's going to come into the chloroplast. Within the chloroplast, you have the thylakoids. Okay? So here's our individual thylakoid. And inside that thylakoid membrane are photosystem 2 and photosystem 1. Photosystem 1 was discovered first, but it's second in this whole process. We start with photosystem 2. The energy of sunlight is going to hit these little green things. These are called chlorophyll. Not chlorophyll, but chlorophyll, right? So it's going to hit chlorophyll, and the energy of that sunlight is going to move from chlorophyll pigment to chlorophyll pigment until you get to a special kind of chlorophyll called P680. You need to know that, P680. Photosystem 2, its acceptor right there is called P680. That's the special chlorophyll there, the primary chlorophyll. The electrons from P680 are going to get excited. That's what this arrow is representing. The electrons are going to get excited, and they're going to move to what they call the primary electron acceptor. Okay, that's what this blue square is representing. Okay? And then those electrons are going to flow down an electron transport chain, similar to what we saw in a cellular respiration. Those electrons are going to move from molecule to molecule in the electron transport chain. So this molecule is going to get reduced, and then it gets oxidized by this molecule, and so on. And the energy of those electrons moving down the electron transport chain, the purpose of that is to pump hydrogen ions from the stroma, the cytoplasm, if you will, of the chloroplast into the thylakoid space. Okay? And then this gradient of hydrogen ions is going to be used in the ATP synthase here, this purple molecule there, and these hydrogen ions are going to flow like a wind turbine. The hydrogen ions go from where there's more of them down through the ATP synthase, like the wind going through a turbine, and the energy of those hydrogen ions going from high back into the stroma where there's a low amount of them, the energy of them going through the ATP synthase makes your ATP. Okay? And then that ATP feeds into the Calvin cycle. But I told you in that last slide that we need ATP and we need NADPH. So how we make NADPH, that's where photosystem 1 comes into play. So those electrons, as we go down through the electron transport chain, those electrons are going to replace the electrons that are being lost from, you have the same process going on in photosystem 1. You have light also exciting the chlorophyll pigments in photosystem 1, and the same thing, it's going to pass it on to, but instead of it being P680, it is P700. So the electrons from P700 will go up to another primary electron acceptor there, and then down a second electron transport chain. And this time the energy of the second electron transport chain isn't used to make ATP, it's being used to make NADPH. Okay, so 2 electron transport chains. A side note, how we replace the electrons that were lost from P680 is through the splitting of water. So this is why the plants need the water. The water is basically a donator of electrons. These electrons from water are replacing the electrons lost in P680, and in the process of that replacement of electrons, you're going to create oxygen. And that's the same oxygen that the plants release and that we then use, and plants use to breathe. Okay? I know there's a lot going on there, but understand that story. It's a story you can understand. Okay? All right. So then moving on to 8.3, which is the Calvin cycle. So that ATP and the NADPH that we just made in the light reactions, we're going to use the energy stored in those molecules, stored in the ATP and NADPH, to then make our sugar. Now, a fancier way of saying make that sugar, what we're really doing is we're reducing CO2. So what I mean by reduce, we talked about, I told you about oil rig. Oxidation is the loss. Oxidation is loss. It's the removal of electrons. Reduction is gain, the gaining of electrons. So when they say we're going to reduce CO2, what we're saying is we're basically going to add a bunch of electrons to CO2. And what we mean by that is we're going to basically add a bunch of carbon to hydrogen bonds to a collection of CO2 molecules. Now, this is going to then create stored energy. By adding a bunch of carbon to hydrogen bonds, that creates stored energy. So C to H equals carbon to hydrogen bonds equals stored energy. The reason for that, and you don't have to know this in this level of detail, but just if you're wondering, why does carbon to hydrogen equal stored energy? Anybody remember that? No, it's been a little while. Why would adding a bunch of carbon to hydrogen bonds to a molecule represent stored energy? It had to do with electronegativity. It's fundamentally what it came down to. Yeah, go for it. Say again? It's nonpolar, yeah. Nonpolar bonds represent stored energy. And so because carbon and hydrogen have similar electronegativities, they want electrons the same amount. There's basically, these electrons are shared in that bond more or less equally. And that represents stored energy. Anytime electrons go more to a more electronegative atom, that's then the removal of energy. And that's kind of confusing. It's okay. It's not, you don't have to know in that level of detail, but that's what's going on here. We're going to add a bunch of carbon to hydrogen bonds to a collection of CO2 atoms. And I'll show you what that looks like when we get into the actual cycle. And then we're also going to regenerate those starting materials that we use, the NADP+, so they say starting material, we're going to regenerate the NADP+ and the ADP that we had used, that we used to build the ATP and NADPH. Okay? All right. So yeah, CO2 is going to enter that Calvin cycle. We're going to use the energy stored in ATP and NADPH to build our sugar. And specifically, that sugar that we build that directly comes from the Calvin cycle is glyceraldehyde 3-phosphate, G3P. Okay? And now this is kind of interesting. To make one G3P, one single G3P, we have to do the cycle 3 times. And they say we have to fix 3 molecules of CO2. This will make a little more sense when I actually show you the cycle, I think on the next slide. But keep that in mind. To make one G3P, you have to do the cycle 3 times. All right. So There's 3 parts of the Calvin cycle: the carbon fixation, the reduction, and regeneration of the CO2 acceptor. You don't need to know like all of the individual reactions in the Calvin cycle, but know that these are the 3 steps of it. We're first going to do carbon fixation, then we're going to reduce, and then we're going to regenerate the CO2 acceptor. And so let me show you what I mean by all those here. So here's the Calvin cycle. Now, fortunately for you, nowhere in I got an undergrad in biology. At no point in my career, I guess you could say, my academic career, did anybody make me memorize this cycle. And that's good, because when I showed you the reaction to cellular respiration, I said if you do biochemistry, you're going to have to memorize those at some point. I think really, unless you study plants, I don't think anybody's going to make you memorize. First you have 3-phosphoglycerate, then you have 1,3-bisphosphoglycerate, and that goes to, you don't need to memorize that in that detail, but know the different phases, right? Phase 1, phase 2, and phase 3. That's what I want you to know. So in phase 1, it's called carbon fixation. The name of it actually kind of makes sense, because what you're doing is you're taking a carbon-based compound. Each of these are representing carbon atoms. You're taking this carbon-based compound, ribulose bisphosphate, called RUBP. RUBP is a 5-carbon molecule. I want you to know that. 5-carbon molecule. We're taking a 5-carbon molecule and using the enzyme Rubisco, which, fun fact, Rubisco is it's a protein, and they think it's the most abundant protein on planet Earth, because there's so many plants in the world, and this reaction is so important. There's, it'd be the most abundant protein. Yeah. Anyways, I thought it was kind of cool. But all right, carbon fixation. You're taking the carbon in CO2. So these are CO2 molecules, and you're going to attach this carbon on CO2 onto RUBP. And in the process, you're going to make a 6-carbon molecule. This 6-carbon molecule is very short-lived. So here's the carbon from CO2. It's attached right there. So we make a 6-carbon molecule. That molecule is very high energy and unstable. It's going to break down into 3-phosphoglycerate. Sometimes you'll see that called 3-PG. Again, don't need to memorize the names of things. Just know it gets rearranged, and then it gets cut in half to make 3-phosphoglycerate. Okay? I'll explain these numbers in a second. Just let me keep going with it. This 3-phosphoglycerate, we're going to invest ATP. I'll get into the numbers in a second. We're going to invest ATP and attach a phosphate group to it, making 1,3-phosphoglycerate. Where the 3 comes in is this phosphate group, 3-phospho. The phosphate is attached to the 1, 2, 3 carbon. And then because we attach another phosphate group using the ATP, we attach a phosphate group to the 1 carbon. That's why you get the 1 now. Bi is referring to 2. There are 2 phosphates. Now we enter into the reduction. Where the reduction comes in, I told you at the beginning that we're adding a bunch of carbon to hydrogen bonds. Where the hydrogens are coming into place, I showed you where the carbons are coming from. The carbons in the carbon to hydrogen bond is coming from carbon fixation. There's the carbon portion of that. Where the hydrogen comes in is from NADPH. The H of NADPH is going to get attached to our 1,3-bisphosphoglycerate in a reduction reaction, right? The gaining of electrons by adding the carbon to hydrogen bonds, and that's going to make G3P. And G3P, that is the output of the Calvin cycle. Okay, G3P is what we're making. Now, phase 3, phase 3 is regeneration. We have to regenerate that RUBP, because that RUBP started this whole cycle. So what we're going to do is basically we're going to have to take some remaining G3P and recreate RUBP. So to understand this is where the numbers come in. I told you that to make 1 G3P, you have to do the cycle times 3. In other words, you're taking 3 CO2s, and we're starting with 3 RUBPs. We're taking 3 RUBPs and we're combining 3 CO2s. Let me use a different color for that. So we're doing this cycle 3 times. That's why there's the number 3 right there. So you're going to take 3 CO2s, 1 CO2 per RUBP, per 5 carbon RUBP, and you're going to make 3 of the 6 carbon sugar here. 3 of those. And then I said this gets cut in half to 3 PG. So you're making 6 3 PGs. So if you're counting carbons, here you add 3 times 5 is 15 carbons. We're then adding 3 CO2s, so that's 18 carbons. And then 3 times 6 is what? 18, so you're keeping track of the carbons. You have the same, the whole class just like 18. So we're keeping track of the carbons, right? And then you're gonna split this into 6 3 PGs, 6 times 3 is 18. So we're not losing any carbon atoms here, right? We're just rearranging the carbon atoms. And then we're making, then you have a 6 here because all you're doing was just adding a phosphate from ATP. And then we're gonna add your hydrogen from NADPH, and you're gonna make 6 G3P. Now, this is really like, keep track of this. It's not that complicated, but like, follow me here. Only one of the 6 G3P is actually gonna be used as like actual net sugar that you're making. Because then 5 of the G3Ps are gonna be reused to remake your 5 RUBPs, or I'm sorry, the 3 RUBPs that you start with. Okay? It's a little weird. What I want you to think of is 1, 3, 6, 9. So you're making 1 G3P. To make that 1 G3P, you're gonna need 9 ATPs. You will need, to make this 1 G3P, you're gonna need 6 NADPHs. My spatial awareness is not the best. 6 NADPHs and 3 CO2s. So this is like kind of like as far as like where a lot of the test questions coming in, inputs, outputs, location. So output, you're making 1 G3P. To make that 1 G3P, you put in, so here, let's do this. This is an output. That's an output. You're gonna put in 3 CO2s, 3 NADPHs, and 9 ATPs to make that 1 G3P. Okay? If this whole cycle is very confusing, I really like the worst case scenario, understand the 1, 3, 6, 9. Okay? That's kind of where a lot of like, for your test on Monday and the AP exam, that's really where they're going to be asking a lot of questions. Okay? Where some of like the more complicated questions get in, that's where they might ask you about phase 1, phase 2, and phase 3. But those would be like the more like challenging questions. Okay? Questions, yeah?
Can you go over the inputs and outputs one more time?
Yeah, okay. So there's only one output really. The main output that you're worried about is G3P. To make that 1 G3P, you need to invest 3 CO2s, right? There's the 3 CO2s. You need to invest 6 NADPHs, that's represented by this step. The NADPHs is the reduction phase. And you need to invest 9 ATP. The ATP get invested in 2 different spots. One step is invested in the carbon fixation part, 6 of them here, and then 3 of them in the regeneration of the RUBP. Okay. Now, sometimes you'll see as part of outputs, you'll see here, let me get this erased. Sometimes you'll see as part of outputs, you'll see, if I go back to this slide, you'll see NADP plus and ADP. So here are the outputs, G3P, And then technically you are also, as an output, you're regenerating the NADP plus and ADP. You could view that as an output. And then the inputs, as I said, was inputs are 3 CO2s, 6 NADPHs, and 9 ATP. Where the ATP and NADPH came from was the light reactions. The CO2 comes from the atmosphere, and then you're just making 1 G3P. Okay? Hopefully coming back to this slide, it puts in a little more perspective. Okay? All right, I'll keep moving. Oh, I forgot to mention this. The Calvin cycle is an anabolic reaction. Remember, you had catabolic and anabolic. Catabolic, cats break things, right? They break things down. Anabolic, anabolic steroids are like building muscle, or like Anna builds things. If you have a friend named Anna that builds people up, you know, you can think of it that way. Anabolic, you're building up a larger molecule. So it's an anabolic reaction there. All of this here, this is really just in words what I was going over. If you wanted to, that helps you to read through it. So yeah, there's that slide again. All right, so that's really about it. I want to show, this is a good slide to kind of bring everything into perspective. So let's walk through this, and a lot of it I've already have shown you. But like think of a tree. The water that we need in our light reactions comes from the soil. Bless you. And ultimately that came from like rain. You know, the water cycle, everyone's favorite. Everybody loves a water cycle, right? The water is going to come up. It's going to travel into our leaf. The CO2 that we need in our Calvin cycle, that CO2 comes from the atmosphere. The oxygen that's produced whenever we got to regenerate those electrons, that's going to get released into the atmosphere. Remember the leaf? The leaf is actually pretty complicated, right? There's a lot of different tissue layers in the leaf. Well, the mesophyll cell in the leaf is the main types of cells in the leaf that are going to do photosynthesis. So zooming in on the chloroplast of a mesophyll cell, that's where the reactions are happening. In the light reaction, you have photosystem 2 first, then you have an electron transport chain, photosystem 1, and electron transport chain. The purpose of all of that was to make ATP and NADPH, and we do you need 9 ATP, 6 NADPH, and 3 CO2 are going to feed into the Calvin cycle. Those 3 CO2, they're going to meet up with 5 RUBPs, right? And then we're going to make 6 of our phosphoglycerates. And then you end up making 6 G3Ps, but only one of them, you have to minus 5 of those G3Ps to remake your, I'm sorry, this is not 5, this is 3. Okay. 3 RUBPs, you only net 1 G3P because 5 of them were used to make the 3 RUBP. Remember RUBP, if we think of it as counting carbons, that's kind of something they want you to keep track of. RUBP is a 5 carbon sugar. CO2 is a 1 carbon sugar, but there's 3 of them. So this is, 3 times 5 is 15, plus another 3, that's 18. 3PG, that is a 3 carbon sugar, but there's 6 of them, so that keeps track of the 18. And then you get out a G3P, that's a 3 carbon sugar, you get out 1 3 carbon sugar. And then you're also going to get out, you're going to regenerate the NADP plus and the ADP. Okay? So there you go. That's kind of the simplest way I could kind of go over photosynthesis. Real quick, 8.4. This is technically part of 8.4. It's really just kind of big picture talking about photosynthesis. I've already said most of it. Remember the energy that sugar that the plant's making. It's going to be used. Half of it is used as fuel. Half of the sugar made is used as fuel. The other half is going to be used to then make other organic molecules the cell needs, whether that's proteins, maybe some fats. So the plant's going to use that basic G3P, that sugar that you make, to then build other things. Okay. This is important. The plant's going to take that G3P that it makes, and it's going to, in order to transport that G3P throughout the plant, it needs to convert it into a different kind of sugar called sucrose. Anybody know what sucrose is? Like sucrose, aka what? It's
Starch.
Not really what I'm looking like when you think of sucrose, what do we think of?
Just like sugar.
Just sugar, yeah. It's aka just like your basic, when you're putting sugar in your coffee, it's sucrose. So anyways, I thought it was kind of interesting. So aka just your normal sugar, table sugar, I guess you could say. If the plant has excess sugar that it doesn't need to like run its cells, we store like excess energy as fat. The plant will store that excess sugar as starch. And a lot of that's going to get stored in the fruits and stuff. That's why the fruits will taste really sweet. It'll also store them in like roots, tubers, seeds, stuff like that. So, and then that oxygen gets put in the atmosphere. And yeah, that's really it for the notes. So yeah, let me get this stopped here.